The tree of life was once imagined as a grand orderly diagram, branches diverging cleanly from a single trunk. But modern genomics reveals a different picture. Between the branches run invisible threads, strands of DNA traded, borrowed, or stolen. Life's tree, it turns out, is laced with bridges. This hidden traffic is horizontal gene transfer, the movement of genes between organisms that are not parent and offspring. In microbes, it is not the exception, but the rule. Through this process, bacteria and archaea routinely acquire genes from neighbors, sometimes even from distant domains of life. A microbe's genome is less a fixed identity than a portfolio, constantly updated with new instructions. One of the clearest examples lies in Escherichia coli. Nearly one-fifth of its genes appear to have come from other species. The deadly strain O157H7, known for causing outbreaks of foodborne illness, contains more than 1,300 genes absent from the harmless laboratory strain K12. Those borrowed genes encode toxins, secretion systems, and entire metabolic pathways. In evolutionary terms, E. coli did not just mutate, it reinvented itself through acquisition. The methods of this gene exchange are as diverse as the microbes themselves. Plasmids ferry DNA from one cell to another through direct contact. Viruses called bacteriophages inject foreign genes as part of their infection cycle. Some bacteria absorb naked DNA fragments drifting in the environment, a process known as transformation. The results are immediate and profound. Antibiotic resistance, novel enzymes, or entirely new ecological roles can appear in a single generation. This exchange blurs the boundaries of species. In hot springs where bacteria and archaea coexist, up to 20% of the archaeal genome may be bacterial in origin. Genes encoding light-driven proton pumps, proteorhodopsins, leapt from salt-loving archaea to marine bacteria, giving them a new way to harvest sunlight. Even eukaryotes are not immune. Our mitochondria are the descendants of bacteria whose genomes still whisper inside every cell. Scientists can detect these foreign genes by their accents. Sections of DNA with unusual GC content or distinct codon usage betray an alien origin. Clusters of such genes form genomic islands, blocks of DNA that often carry traits for virulence, symbiosis, or resistance. These islands are the footprints of ancient transactions, records of microbial diplomacy written into chromosomes. Such trade reshapes evolution's narrative. Instead of a slow vertical climb from ancestor to descendant, microbial evolution looks more like a web, dense, tangled, and self-reinforcing. Ford Doolittle once redrew the tree of life as a swirling network of connections, each line a reminder that evolution is not just inheritance, but exchange. For pathogens, horizontal gene transfer is a survival strategy. By swapping surface proteins, they evade immune detection. For environmental microbes, it is a means of adaptation, sharing enzymes to digest new nutrients or survive toxins. Even in soil, genetic packages hop between bacteria, allowing entire communities to evolve collectively. In this light, innovation is not just invention, but conversation. Microbes do not merely compete, they communicate through genes. Each transfer is a message, sometimes destructive, sometimes creative, but always transformative. If vertical inheritance writes the story of descent, horizontal transfer writes the story of connection. Together, they make evolution not a linear history, but a living network, one that stretches from ancient oceans to the cells in our bodies today.